Okay, let's start, okay? So, ladies and gentlemen, if you have my notes, okay, take this out. The importance of water. You know how teacher tell? Because this is not importance of water in your daily life. So don't tell me about washing lah, cooking lah, okay? It's talking about cell in your body, each cell. What does it need water for? Okay, so I put it like this. I put it in this way so that it's very easy for you to remember. You forgot to unmute yourself. Okay, the first one is to maintain the body temperature. So our cell will use it to distribute heat. High surface tension and cohesion. You know what's cohesion? Co is like cooperate. So the water particles will cooperate and pull each other up. Unbroken columns through the vascular bundle in the cell. We call it the xylem. That is cohesion. That's why we need water. Okay, otherwise how water can move? Anti-gravity, correct? It also have high surface tension that the animals or the insect can stand on the water, can move like water glider, you know, the stuff. Lubrication, okay, that is for you to swallow your food easily, more moisture, right? Got mucus, can make mucus. And in our synovial fluid on our joint, our body joint, between the two bones, so that we don't get crack, oh, every time we use, then a lot of friction already. So that's lubrication. And then we have moisture in our alveoli, okay? And this is our homeostasis, right? Correct? In our osmotic balance. So that we can maintain this concentration of salt in our body. Okay? Support of protoplasm. What is protoplasm? Who knows? Dorothy, you know what is protoplasm? Hi, Dorothy. What's protoplasm? No? Julia, what is protoplasm? Who knows? Type on the chat. Ayo, Arun. Good job. Arun says cytoplasm plus nucleus. Correct. So it's the majority part of your cell. Understand? Cytoplasm, you know, is to have all the biochemical processes inside your nucleus. So it's a support to the cell. And it's also to use to do lysis, biochemical, right? Hydrolysis, breaking all the food that you eat so that your body can absorb when they are in smaller, simpler molecule. And last is a transport medium. 90% of our blood plasma is actually water, so they can carry all our hormone, our heat, our waste product out from our body. So for those who think, wow, very hard to remember, teacher. Oh, I also can't away one. Okay, but a bit 18 SX lah, yeah. So make sure your mom is not listening, Dorothy, Okay, so the first part is we talk about wow, the body temperature very high tonight. When I say tonight, right, all the boys will wake up one. And then right, space tension, very tension. Don't know can perform or not. So you went to buy a bottle of lubricant, all right, to increase the moisture, right, to maintain the rigidity. Then the hand comes ready for support, right? So you know lah, when the sperm come out, right, the biochemical reaction. So you got to transport, you need the ovum, right? So transport. Done already. All the keyword, right? All that things go in the brain easily, right? Then you can answer. Now if I ask you, I think you can remember already. You see, how I do, let's ask Nimalan to try first. Nimalan, a very good boy one. See, he remember. Give me first two, Nimalan. Think first. Think back.
Hello, teacher.
Tonight very hot. Hi.
First one is to.
Tonight very hot, very hot.
I think to maintain high surface cohesion.
Don't kanchiong. Tonight very hot. Reduce what? Body heat first. Correct or not? To distribute the heat in our body, isn't it? In a cell.
Yeah.
Yeah. Then only what?
High surface cohesion.
Yeah. No moisture yet. High surface tension.
No,
High
cohesion.
tension.
Yeah, high tension and cohesion.
High tension. You tension lah. Water is what? Surface tension. Can you use the keyword?
Yeah, surface tension. Sorry, this is
I told you he good boy one. He cannot remember all the bad things. Okay, now I call the not so good boy one can remember one. Okay, Tiba. Continue two more.
Lubricant.
Oh, lubricant. Then
What a question and tension.
very good. Maintain what? Turgidity.
Turgidity.
Yes, turgidity. Okay, now after maintain turgidity, okay, what else? Two more. Let's ask who Azra, can remember or not? After Zahid, your turn.
After that, the last one was the GT, right?
Yes.
Then the hand.
Hand
Support.
what? Very good. Support, especially for those hydrostatic skeleton one, understand? That's the support.
Yeah, understand.
So don't only talk about our own.
Support.
Must think of animal, must think of plants. Then only it is the function of water in cell. Okay, one more. Support. Then
Support, and then
what come out? Shoot out already is what process?
Biochemical process.
You are right. Zahid, last one. Your turn. After biochemical process, then
Transport.
good job, like you guys. See or not? Very easy, right? Biology. Study with teacher Joyce. Okay. So well done. Now we go to carbohydrate. Okay. Yeah, very naughty. Yes, I know, right? Okay. So if you look at this, the first one, we talk about carbo, and I taught you before, right? This is my 2GF, two girlfriend. Life is very simple. You only got two girlfriend. Life become complex if you want to buy them baju. You don't know their size S, M, or L. Then poly means many. After many consideration, you just don't buy gift lah. Okay, Christmas now. So bring them to GSC. Long time never see watch movie. Okay. Oh, can bring two girlfriend watch movie together. Can I think the ear also drop already halfway before going to the cinema. Okay. All right. So the 2G is glucose and galactose. F is fructose. SML is your maltose, sucrose, lactose. GSC are all the poly, means big structure. Yeah. Now these are the three types of carbohydrate. So put it very easily. I told students already. Okay, from the beginning, you all know. Some of you follow, some of you don't know, forgot already. Let me go through. So maltose is actually glucose, the basic one, plus another glucose, because it's called disaccharides. Di means two. Okay. Sucrose is still glucose, but plus what? Plus fructose. Why? I put that there. This is fruct, is like fruit. SUC is suck fruit juice. Then lactose one is also glucose, but now you add with galactose. What am I spelling? Galactose. Because why? Because galactose inside got the word lactose ma. So lactose and lactose combination. Okay. Starch, we have it in plants. We know, right? Plants will store food in a form of starch, but human animals, we will store as glycogen in our liver. Okay, tak cukup, only turn back by your insulin. Cellulose is the cell wall. So yes, again, it is the plants. You don't have cellulose, sorry. Okay, so this is the different type of carbohydrate. You will write down? I wait for you. Anyone wants? Okay, write down, yeah? For those who wants to remember it. Yeah, that's how we do it. So very easy. Yeah, I see thumbs up means you need. You need, then I will continue, let you. Ayo, so cute lah. I love all the Christmas. Thank you, Joash. Thank you, Wei. Thank you, Sarah. Lionel, all remember to put Christmas for me, oh. Ayo, I feel so Christmassy now. Yeah, I also got. Okay. Go on, ah. So this one I skip, ah. Okay. So when we see monosaccharides, okay, we only study about the simple sugar first. All right, simple sugar first. Then we talk about glucose. You see G, it comes from grape one. Fructose is fruit, ma. Can you see all the fruits got fructose? Galactose, anything got lactose. You remember lactose intolerance? So that's related to milk. Okay. We use Benedict B. Benedict is what color? Blue. Benedict is one of my girlfriends when I was little in kindergarten. And I remember she don't like blue, she like red. So it becomes brick red. It will change. Yeah, copper 2 become copper 1. Copper oxide is red. So what we use it for is when the long chain of these monosaccharides, they link to the protein or the lipids. Then it becomes the tagging. You remember the tagging on our protein? Our pore protein. We got glycoprotein and glycolipids on our pore protein that links it. Yes, that is the tagging for on our plasma membrane. Now this one just now I told you already. So when you take a big structure, okay, why I say big structure? Because maltose is a disaccharide, right? And when you want to break it down, it's the opposite. So opposite is called hydrolysis. Lysis is again, it is to break down or to burst. So the opposite is we add water. You take maltose, you add water. You can draw another arrow on the opposite side. That is hydrolysis. You understand? And when you take the two glucose put together, you want them to chantong, mix together, you need to take one small molecule out. And that is also what we call condensation, because you release the water out, remember. Okay, so your SML comes from where? Sucrose comes from sugarcane. Again, lactose is a milk. It can be mommy's milk, mammal's milk. And malt, you know what? It is the one that make beer or Guinness malta or malt like your Milo. Milo is a malt drink. That's why I give you a lot of energy to do your sports. Now you know lah why Milo sponsor all the sports event. Okay, so this part is even easier. I say we got S, M, L. When you buy the wrong baju for your girlfriend, M and L are called reducing sugar. Means these two sides can still reduce to S. Can you remember that? And S is the smallest size in my shop. Sorry, I don't have extra S. So it's called non-reducing sugar. That's all you need to know. Don't worry about the others. Okay? So you see, so easy, right? So now I tell you, glucose comes from where? G. You remember grapes. Galactose? Oh, milk. Lactose? Milk. Right? How hard is that? Just a little bit of extra effort only. Okay? You can remember all the details already now. Okay. So now we go to polysaccharides, like just now I say, right? Polysaccharides, the starch is stored as storage. for plants. And therefore, we find it in potatoes lah, in corn lah, okay? Eat already very kenyang one, right? You eat cornflakes in the morning, right? You can take a baked potato already, is one meal. Or French fries, you eat already very kenyang, right? It's because a lot of starch. It's same value like eating rice, same value like eating porridge or noodle, okay? Because noodle also made from wheat flour, wheat, gandum. All these are starch, okay? So how do we test starch? We use iodine, and iodine is brown in color. It will turn blue black, okay? But I said glycogen is for animal. There's one more thing called yeast. Yeast also stored as glycogen. You can find glycogen in yeast. But cellulose, you see the word C-E-L-L? We have it in cell, we have it in wall, cell wall, all got cellulose. So because they're so long, right? You want to break them down? Can do hydrolysis. But this time, because they are more stubborn, you must put some enzyme. Otherwise, it won't happen. So enzyme to break it down. Okay, clear? Okay, next one. So like just now I said, release out water called condensation. Add water to break out something, we call hydrolysis. Okay, so this is what it means. Okay, now I go to protein. Okay, protein also last time I teach you, I teach you before and then I protein. So amino acid is the smallest. So you took amino acid and amino acid, you put together, boom, become dipeptide. Dipeptide, add another one, boom, become what? Let's ask Wing Yan. Dipeptide, add one more amino acid, called what? Wing Yan there, right? I remember. Got. Wing Yan, on your mic. Talk to teacher Joyce. Di is 2, right? I add one more amino acid at the back here, like this. Then call what again?
Polypeptide.
So fast poly, Not yet, not yet, not yet. Di become what first? 3 is what?
Tripeptide.
Yes, it's called 3 is tripeptide. Very good. So you ask teacher Joyce, teacher, then until when only I call polypeptide, correct or not? Yeah, that's why I want you to feel it. Okay, so let's see this. Okay, we have 2 types of protein, and you can see if anything lesser than 75 here. Now, this is what they always ask. Okay, it's still a long chain of protein and it's still in a straight line. So consider small, okay? Dipeptide, tripeptide, until a lot, poly, but must less than 75 only is a straight line. Okay, now, so most of the things that we eat is a straight line, right? Okay, so now let's see, we go back to this one first. I want to start from here. So we have essential amino acid, means you must eat from your diet one. Essential means important for you, okay? 9 types is important. 911, can you see? 11 is non-essential because why? We can take from whatever we eat and make this 11 type. Yeah, so we only need 9. So who are they? Well, we're going to see the names a little bit. Then first class protein is always related to the animals because animals has all the 9 types of essential amino acids we need for our body. Be it you say, teacher, what if I am a Muslim? I never eat pork, right? Will I get also all the 9 types? The answer is yes. Okay. If you eat fish, yes, only fish also can. You understand what I mean, right? But if you are vegetarian now, okay? This is second class because plants do not contain all the essential one. Now you know the difference. You can be eating a lot of different beans, but you still don't get enough protein compared to a person who only eat fish. Okay, so in your body, there are certain things that's lacking one if you are vegetarian. Any vegetarian here? Can I see show of hands? No, no. No. Oh, all carnivorous here, huh? All eat meat one, huh? Not even omnivorous, huh? Some don't eat vegetable at all. I know one. I know you. Okay. So don't have balanced diet, okay? You're big enough already. Okay, so these are the essential ones. They never ask you an exam one. I just give you the list so that you know if you're buying a protein shake for people who muscle building on. All right, you want protein. Then what kind of protein you're drinking? I'll go and find. The list is on the internet. Don't go and buy something that has non-essential one. Papaya one, go and buy for what? Okay, so you need all these others. Then only it will help you to build the non-essential ones. Understand? Okay, so let's look at the 4 types. I like to use this to see. And I like to use this to read. I use this to see first, okay? So first, it is a long chain. The long chain must be fewer than 75 because if more than 75, they will turn into folding stuff like this. Either is alpha helix. Alpha helix is like a spiral staircase, turning, like your DNA. Or it will be a beta sheet. It's like a paper, okay, folded up and down like this. Okay, of course, it's a sharp one. All right, yeah, so that is beta pleated. So either one, also it's called secondary structure. Secondary level. We have this primary structure, secondary structure, and the tertiary is the third one. So look at this. This is one folding. The one folding here match up or turn into tertiary. Yes. And quaternary means I must at least have 4 of the tertiary combined together, like this one. Red one consider as 1, darker one is 2, brighter one is 3, but got shadow. White color one, no shadow, is 4. Got 4, then it's becoming quaternary. Okay? Now, let's look into our body. Okay, everybody look at my videos, okay? So primary is the one we eat. Then we break into all the amino acid, right? Then we're going to make what? Wow, our skin. Our skin got collagen. Our nail got keratin. It's all the top part. That is secondary, okay? Okay, now I correct. I dig a hole. I open up. What I see inside is your tertiary. A lot of things floating around, right? We got enzyme floating around. We got hormone floating around. We got protein floating around. We got antibodies floating around. Okay, so it's like you open a hole and you see, oh, lot of things. Oh, who carry them? Oh, this is your red blood cell. So the quaternary one is the 4 chain that has to put up together. That is a quaternary. Okay, and hemoglobin is the 1,4. So you could see here, I put here 2 or more tertiary. So in our syllabus, they always ask is hemoglobin only. Okay, this one is what flow in your red blood cell. So where is the right with that? From the skin, then go in, what is floating, then is what is carrying it. Done. 1, 2, 3, 4. Okay, all right, now we go to lipids. Lipids, the main thing is CHON. Why I say CHON? CHO. CHON is your protein. Carbon, oxygen, hydrogen also. Same like glucose, yeah? Same like carbohydrate. Also these 3 only. Protein has N, nitrogen. So lipids is the main type of lipids, like your fats, oil, wax. On the leaf got wax. Those are their lipids. Your phospholipid bilayer on your plasma membrane. Steroids. What are steroids? No need to take one, okay, guys? You have it. You have cholesterol, is a steroid. Boys, you got testosterone. Girls, you got estrogen and progesterone. All these are hormones, right? All these will alter your thinking, your feeling. Those are steroids. So fats are solid in room temperature. Liquid one we call oil. Okay? All right. So highlight those important ones, okay? These are why we need fats in our body. Without it, I tell you, your hormone go haywire. Your period also cannot come. Understand? Cannot be too thin one. Who too thin one here in the class? You will see. I don't know. I don't know who. And I don't need to ask who. You know yourself. Period also not consistent. Why? Hormone inconsistent. Lack of what? Lipids. So must always have balanced diet. Don't scared of fats only. After your body, very pretty, but look like what? Look like a hanger. Hang clothes only. Some place must be big, some place must be small, okay? Must make sure it's balanced. That only is pretty girl. Not pretty hanger. Don't learn the model one. Not nice. Teacher Joyce one is perfect. No need to say already. Correct, boys? Boys. Okay, so now we look at this. This is a structure of a lipid. Lipid has 3 fatty acids and 1 glycerol. So many students don't know what is glycerol. You see, guys, now you're already in Form 5. So easy to teach this chapter already because you learn it in chemistry. Anything all is alcohol. Correct? So alcohol got 1, 2, 3. I remember I asked Dorothy that day, right? Alcohol. Glycerol is actually your propan 1, 2, 3 triol. Correct or not? Why propan? Because got 3 carbon. Why 1, 2, 3? Got 3 OH. Why triol? Got 3 OH law, Teacher Joyce. Understand? So that is the scientific name that you can draw it out one. Write in. Now then where is the 3 molecule of fatty acids? Now look at this. Anything fatty acids is like your carboxylic acid. So you can see here is your COOH. 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 So the 3 fatty acids all release out the H. Glycerol release out the OH. Then they chantom together, become a 1 molecule of fats. And they will throw away 3 molecule of water. Understand? 3 molecule of water come from where? There, here should have another H on because it is carboxylic acid. So H plus this OH. Another H here plus this OH. Another H here should be here plus this OH. So when we form this, we will always throw away 3 molecule of water. Then only it's a balanced equation. Clear, guys? Yeah. So the carbon can be very long. I don't care how long is this carbon. Yeah. The only thing I care is whether they are double bond or single bond between the carbon. Okay, why I say that? If they are double bond, look here, like this. You see the structure can make one koyak a bit, right? Means very easy. We can break it down. So you eat anything got unsaturated fats is considered healthy. So you see this one? Oh my goodness, all the carbon between carbon, all single bond. Means all of them so stable. You think you can break them easily? No way, right? So saturated fats are unhealthy fats. Understand? Write down. This one is unhealthy. This one is healthy. Now, what kind of oil that we eat that is healthy? Anyone knows? What kind of oil you eat, Farah, you consider self-healthy? Farah, no, no.
Plant oils. I don't know.
What? What kind of oil?
Plant oil.
Plants oil, right? Yeah, correct. Plants oil are healthier compared to animal oil. Okay, can you give some example or not? Let's ask Jing Xuan. What are the plant oil that Farah say? You can think of 1 or 2.
Olive oil.
I like that. That's my favorite too. Some more?
Sunflower oil.
I also use that. I use canola as well. Yeah, those are flour. Well, corn is a mixture. Okay, corn has some unsaturated as well. Same as coconut. It depends. They take the santan or they take the virgin coconut oil. Got different type of oil, right? Just like our palm oil, also the same. They got combination one. So not all plants oil, the one that I mentioned, especially coconut, corn, and also the peanut oil. Peanut. Don't need so much peanut. high cholesterol also. And also our palm oil has a combination, okay, of unsaturated and saturated oil. This one. Yeah, you all can go and Google. You don't have to believe me, right? Because in our books, we say that, oh, palm oil is super good, very good. Of course, we are Malaysia, largest exporter. I tell you my palm oil not good in my textbook. And who's going to buy all? Correct or not? So you must understand the difference, okay? So what do we use lipids for? Well, for the plants, even for the birds, their wings have wax so that it's not wet when they fly. The bee will use wax to make the beehive. Now, these are all natural stuff, guys. Okay, so it is the cuticle of the epidermis on all the leaves and the fruits and the seeds. They come naturally. Okay, of course, we have a lot of phospholipid bilayer. That is where the lipids is in our cell as well. Because steroids, just as I said, steroids are usually the cholesterol, all the hormones. And we use all this to, is a precursor, means the beginning, to synthesize this steroid. To make hormone, we need fats. Yeah. Okay, so let's look into the two types of fats. We have saturated and unsaturated, like I said, right? Saturated one are usually from the animals. Unsaturated are usually from the plants. Okay, done. So now we're going to watch a very easy chemical test, food test, so that everybody can remember what is happening, including DCPIP, okay? Okay, let me prepare this. For those who have my notes, you want to write down this. You see, right, guys, most of our things are blue color. Benedict B, blue, okay? Burette blue, because has this copper sulfate. DCBIP blue, right? Emulsion is used ethanol. So this one only become cloudy. Okay, starch you all always do one. So it's blue black. Again, it's brown. Iodine is brown. Yeah. Okay, one of it we studied last time is Milan solution. Milan will turn brick red if there is protein. But we no longer like Milan already because Milan is not accurate. Anyone remember why Milan is not accurate? Why we prefer burette test? Who can tell me? Anyone? Can on your mic, can you help me? When we are lower secondary, we always use Milan. We never mention about burette test one. Remember? Okay, after I explain, I'll let you watch the video first.